In this session, we are going to study basis of classification. Under the basis of classification, I am going to explain the following points in brief. 1. Symmetry in animals. 2. Diploblastic and triploblastic organization. 3. Coelom in animals. 4. Segmentation in animals. 5. Notochord. 6. Classification of animals. Even though animals are different in structure and form, there are fundamental, common features. For example, arrangement of body cells, symmetry in body parts, nature of coelom, patterns of digestive system, types of circulatory system, and reproductive system, etc. These features are used to classify animals into various levels. Even though all animals are multicellular, the arrangement of cells are not in the same pattern. For example, in sponges, the cells are arranged loosely and form cell aggregate body pattern, and is known as cellular level organization. Some division of labor occur among the cells. In coelenterates, the arrangement of cells are more complex. Here, cells performing the same function are arranged into tissues. Thus it has tissue-level organization. In Platyhelminthes and other higher phyla, tissues are grouped into organs, and hence, organ-level organization is found. Each organ specialized for a particular function. In annelids, arthropods, mollusks, echinoderms, and chordates, organs are associated into organ system to perform specific physiological functions. This type pattern is called organ system level organization. Organ system shows various complexities. 1. Digestive system may be complete or incomplete. If only one opening present that act as mouth and anus, then it is called incomplete digestive system. Seen in Hydra, platyhelminths, fasciola. 2. Complete digestive system. It has both anus and mouth at opposite end. It is seen in higher animals. Variation in circulatory system. 1. Open circulatory system. Here, blood pumped out of the heart, and the cells and tissues are bathed in it. 2. Closed circulatory system. Here, blood circulated through a series of blood vessels, arteries, veins, and capillaries.
Symmetry in animals. Animals can be classified, based on the symmetry, into 3 categories. They are: 1. Asymmetrical animals. In these animals, any plan, passing through the center, does not divide them into equal halves. Examples: peripherans and gastropods. 2. Radially symmetrical animals. In the say animals, any plan passing through the central axis of body divides the organism into 2 identical halves. Example: Hydra. 3. Bilaterally symmetrical animals. These are animals which can be divided into 2 identical left and right halves in only one plane. Example: annelids, arthropods, etc. Depending on germ layers, animals are classified into 2. They are diploblastic and triploblastic animals. 1. Diploblastic animals. These are animals in which the cells are arranged into two embryonic layers, an external ectoderm and an internal endoderm. Examples: porifera, coelenterates, and ctenophores. In diploblastic animals, an undifferentiated layer called mesoglea is present in between the ectoderm and endoderm. 2. Triploblastic animals. These are animals in which the developing embryo has a third germinal layer, called mesoderm. Mesoderm is present between the ectoderm and endoderm. Examples: Platyhelminthes to chordates. Nature of coelom in animals. Animals are classified into three types, according to the presence, or absence of cavity, between body wall and gut wall. They are: 1. True coelomate. These are animals in which the body cavity is lined with mesoderm on both inner and outer side. These type of animals are called coelomates. Example: annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. 2. Pseudocylomates. These are animals in which the body cavity is not lined with mesoderm, but mesoderm present as scattered pouches in between the ectoderm and endoderm. These types of animals are called pseudocylomates. Examples: Aschelminthes. 3. Coelomate. These are animals in which body cavity is absent. Example: Platyhelminthes.
Segmentation in animals. In some animals, the body is externally and internally divided into segments, with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord. Animals are classified into non-chordates and chordates, according to the presence or absence of notochord. Notochord is a mesodermally derived, rod-like structure, formed on the dorsal side, during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates, for example, porifera to echinoderms. Classification of animals. Kingdom Animalia. Based on level of organization, animals are classified into 2. 1. Cellular level and 2. Tissue, organ system level. Cellular level organization have only one phylum, that is, porifera. Tissue, organ system level are classified into 2, based on the symmetry. They are: 1. Radially symmetrical and 2. Bilaterally symmetrical. Radially symmetrical have 2 phyla, they are: 1. Cnidaria and 2. Ctenophora. Bilaterally symmetrical animals are further classified into 3, based on the nature of coelom. They are: 1. Acoelomate animals have no body cavity. Example: Platyhelminthes. 2. Pseudocoelomate. Animals having false coelom. Example: Aschelminthes. 3. Coelomate animals having true coelom. This includes 1. Annelida. 2. Arthropoda. 3. Mollusca. 4. Echinodermata. 5. Hemichordata. 6. Chordata.